As organisms continue to thrive on this green orb, many creatures living in connectivity must learn to adapt and survive the harsh comings that Mother Nature has presented them. The animal, though big, contains millions of tiny cells focused on supporting the creature to the utmost they could. Unknown to the naked eye, we can see a spectacular organization working in union, all parts playing an important role. Meiosis is very similar to mitosis. However, meiosis is separated into two parts, meiosis 1 and meiosis 2. Both steps have interphase, prophase, metaphase, anaphase, and telophase. Now, in meiosis 1, in interphase, homologous chromosomes are duplicated into sister chromatids, as in mitosis. Then, the sister chromatids are joined at the centromeres. In prophase 1, duplicated chromosomes condense and begin to disappear. The homologous chromosomes pair up and chiasmata occur as chromatids of homologous exchange bits of DNA. This process is also known as crossing over. After that, the nucleus disintegrates. The spindle fibers begin to form from centrioles and attach to chromosomes at kinetochore proteins at centromeres. In metaphase 1, paired homologous chromosomes line up along the cell equator. One homology pair of each chromosome faces each pole of the cell and attaches to a single spindle microtube via kinetochore. Kinetochore proteins are located at the centromere of each homologous pair of sister chromatids. The pole of the cell the homology pair faces will determine which pole it will migrate to during anaphase 1. Which homology pair faces which pole at the cell is a random choice. This randomness is referred to as the law of independent assortment. In anaphase 1, homologous chromosomes pairs separate. Each homology pair randomly moves to each pole of the cell. In telophase 1, spindle microtubules disappear. In each pole of the cell, they each have one set of sister chromatids corresponding to each chromosome in the original cell. An example, each pole of the cell will either contain one pair of the maternal or one copy of the paternal chromosome pair. The nuclei are 2N, however, they can be thought of being N, since they contain only a maternal or paternal chromosome. A nuclear member may or may not appear during telophase 1. The first stage of meiosis 2 is prophase 2. In prophase 2, there are two cells as results from meiosis 1. Each cell contains one chromosome of the homologous pair. After prophase 2 is metaphase 2. In metaphase 2, the two X-shaped chromosomes align themselves along the diameters of the cell. Next is anaphase 2. In anaphase 2, the sister chromatids move to the opposite poles of the cell. Once they separate, each are considered as chromosomes. The fourth stage of meiosis 2 is telophase 2. In telophase 2, the spindle fibers begin to disappear and nuclear membranes begin to form around the separating cells. Here we spot a leafy green out in the wild in its natural habitat. Now I have my assistant here with me to take a closer look at the eukaryotic cell. If you couldn't tell, it's a similar cell to an animal cell. Plants and animals are both pertaining of eukaryotic cells. However, there are some differences. For example, both the animal and plant cells have mitochondria, but only the plant cells have chloroplasts. This is required for the plant cell because they don't get sugar from food, and they need to make sugar by using the chloroplasts and through photosynthesis. Animal cells don't need chloroplasts because they get sugar from the food they eat. Plant cells have one large vacuole, however animals have smaller ones and more. Plant cells have a cell wall and a cell membrane, however animal cells do not have a cell wall. The cell wall allows the plant to have a rectangular shape.
Mitosis is a process where a cell divides into two daughter cells, also known as cell division. In a living creature or plant, mitosis occurs for growth and to replace old and worn-out cells. There are multiple phases in mitosis: interphase, prophase, metaphase, anaphase, and telophase, and cytokinesis. In interphase, the DNA in a cell gets copied to prepare for cell division, which creates two identical chromosomes. In prophase, the two chromosomes condense and form an X shape, while the nucleolus in the nucleus starts to break down. Soon enough, the spindle fiber starts to form. During the early stage of metaphase, the pairs of chromosomes line up on the equator of the cell and the nucleus membrane completely disappears. The chromosomes align in a straight line right-angled from the spindle poles. In anaphase, the sister chromatids are drawn to opposite spindle poles, which means the two duplicate DNA copies are drawn to both sides of the cell and now is ready for cell division. The process of cell division is coming to an end. In telophase, the nucleus membrane and the nucleolus reforms again around the two sister chromatids. Cytokinesis is the process of cytoplasm division.
In the end, we have learned meiosis and mitosis are the basic fundamentals of life, as every living organism is made of millions and billions of cells. Thank you for watching.